All right, my name is Bethany Fowler, and I'm going to be sharing some work that I've done with Michael Newbert and Heidi Sossik's lab at the Woods Hole Oceanographic Institution. We've been studying picophytoplankton responses to seasonal change, so I'm going to start by introducing the picophytoplankton. This is a group of photosynthetic organisms that are classified by their small cell size. They tend to be 1 to 2 microns in diameter. And this includes some of the most abundant primary producers on the planet. The distributions of prokaryotic and Synechococcus are shown on the right here. You can see they're found almost all over the world at concentrations of tens of thousands of cells per milliliter. So the picophytoplankton are contributing greatly to global primary production. They're also expected to make up a larger component of the phytoplankton community with the ongoing changes to the climate. So we're particularly interested in how this group, what role it's playing in the ecosystem and how it's responding to environmental change. So the question here is, how do the picophytoplankton populations respond to environmental variability? And I'm going to be answering it by looking at observations of picophytoplankton across a variety of environmental conditions. So those observations are going to be coming from the Northeast U.S. Shelf Long-Term Ecological Research Site, which is shown by the star here. If we zoom in there, we can see our region has this nice wide continental shelf where most of our sampling takes place. And in particular, we're going to be focusing on this region south of Cape Cod. So to answer my question, I'm going to start by talking about the environmental variability that comes with the seasonal cycles by making use of a stationary observatory that's there year round. And then I'll move on to looking at some cruise data to see how that seasonal response is representative of populations further offshore. So we'll start with the observatory data. The observatory is the Martha's Vineyard Coastal Observatory, or MVCO, indicated by the star here. At that site, we have deployed an in situ flow cytometer, which is sampling the seawater autonomously every 20 minutes and measuring the particles for their fluorescent signatures and estimates of cell size. So from that, we can cluster different groups of picophytoplankton, like the Synechococcus, which is shown in red here, or the eukaryotes, which are in green. And we have multiple instruments that we've cycled in and out so that we have continuous observations that have been going on since 2003. So this is a tremendous source of information about the picophytoplankton population. And we're going to use it to answer this question about seasonal environmental variability. So we're going to start by looking at Synechococcus concentration versus temperature. And here, concentration is on a log scale. So the color is indicating the season. And if we start in the dark blue in the early spring, we can see concentrations of Synechococcus are low and temperatures are low. And then as temperatures increase over the course of the spring, Synechococcus undergoes this really dramatic bloom, reaching its highest concentrations in the summer. And then in the fall, as temperatures decline again, we see a more gradual decrease in concentration back to winter lows. And these are daily averages. So this is a really nice, clean result, but it doesn't provide us with any mechanistic information about why there are changes in population size. And it would be nice to have that mechanistic information because that'll be more useful for making predictions. So in particular, changes in population size are always going to be the result of both cells being added to the population and removed. So in this case, reproduction through division and loss from mortality. And these processes are going to be happening in a much shorter time scale than the seasonal cycle. And in particular, division rate here would be really nice to quantify because not only does it allow us to disentangle the net growth from the loss in the reproduction, but it also provides some physiological information about the Synechococcus. So if they're dividing very rapidly, that indicates that they're pretty happy in their environment, versus if they're dividing more slowly than we know they're capable of, then something might be limiting their growth, and that would be important ecological information to have. So how can we get at that division rate? Again, like I said, we have to zoom in to a shorter time scale. This is a week of data from the Coastal Observatory with picophytoplankton concentration over time. And in addition to concentration, we also have information about cell size. So now the heat map below is showing the size of the population as it goes through these daily cycles, getting larger and then smaller. And these yellow bars here are indicating dawn, as we can tell from the sunlight data that's available at the observatory. So the cells tend to be getting larger while the sun is out and bright, and then they're getting smaller primarily through division. So embedded in this size distribution information is information about their reproduction. We just need to figure out how to extract it. And we can do that using a size structured matrix model. So here's our model where we bin Synechococcus cells into groups by volume, and we allow them to either grow into a larger volume class or divide and produce 2 cells of half the volume, or they can stay in the same class. So this is the transition matrix that corresponds to that model. And of course, the values of the elements of this matrix will be informed by our parameters. And we're going to estimate the parameters by fitting the model to our observed cell size distribution with the forcing of the sunlight data for that day that's available. So each day, we're getting new parameter estimates, a new model. Then we're going to simulate our cells in the model and see how frequently they're dividing. And then we use the division rate of the simulated cells as our estimate for the division rate of the cells in situ. And this works pretty well. Not only are we able to reproduce the size distribution dynamics that we observe, shown by the simulations here, but we're actually getting division rate estimates that are consistent with those that we would get from a dilution series experiment, which is the more traditional way for estimating division rate of these populations. But importantly, our method doesn't require that the cells be taken out of their environment and put into bottles and incubated. It just requires observations of the cells at this really high resolution. So we've done this for 2 classes of picophytoplankton for every day of our time series for the 16 years that we had at the time. And we're going to use this information to now relate the division of the cells to those changes in temperature. So here we're looking at Synechococcus division rates on the top and eukaryotic division rates on the bottom. Both have a similar response to temperature. The seasons again indicated by the colors. So starting in the early spring again, division rates are low and temperatures are low. And we see increases in division rate with increasing temperatures. And this suggests that the populations are limited by temperature early in the spring because as those waters warm, they're able to divide more rapidly. Whereas if we look at the fall, we see that the cells are not able to divide at the same rates that they are for days of the same temperature in the spring. So, sorry, maximal division rates in the summer. But here, importantly, the division rates in the fall are lower than would be expected based on their temperature, which suggests that something is limiting their growth. So if we look at the relationship between division rate and sunlight, we can see there's a nice linear relationship between the two, whereas the sunlight is dwindling in the fall, division rates are again decreasing. So again, this is suggestive that light is limiting growth at that time. All right, so those are our kind of hypotheses that we formed from the stationary observations. And now we're going to move on and see if that's representative of the picophytoplankton populations offshore. And we'll look at this cruise data, which is actually across the spatial gradient. So our transects go from nearshore off to the shelf break. And these are the cruise tracks for the 17 cruises that I've considered for this work. And you can also see the timing of the cruises here. You can see that we have cruises at all different times of year, all seasons, but our temporal coverage is much smaller than we get from the observatory, right? Because each cruise is only a week or a couple weeks long. But we still have flow cytometry data that's being collected from the underway flow through system. So sampling continuously at a 2-minute resolution as quickly as our instrument is capable of. And we're going to use that in a similar way to the observatory. And I just want to highlight here that the cruise data is going to be capturing both the seasonal temperature variability that we saw at the Coastal Observatory and also this spatial gradient that I mentioned. So near shore, we have relatively cold, fresh shelf waters. And then further offshore, we get mixing with the warmer, saltier Gulf Stream. So there's this nice mirroring between the seasonal variability and the spatial gradient that has a similar range in temperatures observed. So do we see the same response offshore as we do at MVCO? As a reminder, here was our nice result about Synechococcus concentration versus temperature. And here is all of our cruise data from all of those samples on the cruises. So they're not daily averages, it's a little bit more variable. But the result is quite similar, right? The relationship between Synechococcus concentration and temperature appears to be consistent for the nearshore populations and further offshore. Except perhaps as we reach these really warm waters that are never observed at the Coastal Observatory. The waters don't get that warm and we see lower than expected concentrations of Synechococcus in those waters. And I'll come back to that. I also want to highlight that if we pick a cruise with a particularly wide range of temperatures, such as AR29 here, we see a quite similar response relationship between Synechococcus concentration and temperature. So whereas over here we were seeing the relationship played out over time over the course of the spring, here we're seeing the same relationship, but over just a few days of the same cruise. So we have this nice space for time substitute where the response of the population is similar, whether that variability is coming from the seasonal fluctuations or a spatial gradient. And here's all that together. So this is including both seasonal variability and that spatial variability. Okay, now let's look at the division rate of the populations from the cruise data. And as a reminder, we needed those high resolution observations of the individual cells in order to get this for the coastal observatory. And we're going to do something similar for the cruise. But because the ship is moving and we might end up with the cruise transitioning between different bodies of water during the course of a day. So we don't want our division rate estimates to be sensitive to that. So rather than using just these 5 dawn to dawn day cycles that we might be able to fit in, instead we're going to use a sliding window approach where we'll fit the model to every 24-hour period of the cruise, starting at every hour of the cruise. So we're going to have some overlapping days, but the result is we get this nice continuous division rate estimate. And we can do things like plot that against the average position of the ship during all those samples, or we can look at the variability in temperature or salinity of those samples to see if we're sampling a single population of Synechococcus or whether we're transitioning between widely different populations or bodies of water. And then we're going to use that to investigate our hypotheses from the coastal observatory. So you'll remember our relationship between division rate and temperature and division rate and sunlight. These are both for Synechococcus here. And these were the climatologies. So that's from our whole time series averaged down to just the each day of the year. But here's the full variability of that data set. So every individual day, and I'm using this because I think it's a more accurate comparison to the cruise data where we don't yet have enough to do a climatology. So you're going to see the division rate estimates for all of those overlapping window days of our cruises. So overall, the relationship between maximum division rate observed and temperature is quite positive and similar to that we see at the coastal observatory. However, the division rates offshore are lower than would be expected based on the temperatures from the coastal observatory. So this could be a result of some taxonomic differences. The composition of the community could be different further offshore than near shore, and maybe they're just not capable of dividing more rapidly. Or it could be that they're limited by something other than temperature. The waters tend to be a little warmer offshore, but they're still not dividing as rapidly as they were near shore. And one hypothesis for this is that the mixing is greater further offshore of the deeper water column, and we could end up with light limitation in the phytoplankton due to that mixing. So even though the waters are warm enough and the sunlight is maybe bright at the surface, they're not getting that sunlight over the course of their whole day because they're being mixed to death. The relationship between sunlight and division rate is more similar offshore to the observatory. So again, we have some days in the spring where there's high light below division rates, and then in the fall, more closely related division rate and sunlight. And here's that same data just plotted over time against the day of the year. So you can see the colored dots are the cruise data. They come in these bars because it's just short windows of time. And the gray dots are the observatory division rate estimates. So we have some higher division rates offshore in the winter and early spring. But as the waters warm up and the division rate is increasing further inshore, it kind of is a delayed increase offshore. So we're not seeing those maximum division rates until later in the summer for the offshore population. And I just want to bring us back to this relationship between Synechococcus concentration and temperature, because I think it's really interesting that this cruise here where we're seeing lower than expected concentrations corresponds to this period where we're seeing lower than expected division rates. And our division rate estimates are independent of concentration. So there's no reason in our methods why the concentration lower would lead to having a lower division rate. But biologically, of course, if the division rate is lower and loss rates are similar, then you'll see lower concentrations. So I think that's exciting and something to explore here. And I'm going to finish with the conclusions here. Just The main one being that the relationship between temperature and Synechococcus concentration is consistent across seasonal cycles, both nearshore and offshore, and that relationship is consistent whether the variability is coming from change over time seasonally or change over space. Secondly, we have this new spatially explicit hypotheses for controls on Synechococcus growth, where before we thought that, or we still think that the temperature is limiting their growth near shore in the winter and spring, but then further offshore, they might be light limited or being limited by the mixing of the water column. So with that, thank you all for watching this video. And if you have questions, email me or I'll take them in the live session. Thanks.